Welcome back to the next webinar in our Plantae Presents Global Plant Science Talk series. My name is Katie Rogers, and I'm your host for today's webinar. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I would like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities we provide at aspb.org. Many of you may have attended ASPB's annual meeting, Plant Biology, in the past. Each year, this event brings together plant biologists to talk about the latest research topics. In 2020, this event was held completely online and we had over 2,500 participants. ASPB's program committee is currently working on making plans for next year's event. The overall theme for next year, is plants powering the bioeconomy. If you have ideas for workshops and would like to submit your ideas, please submit us a proposal by December 4th. Although we are currently planning for a face-to-face meeting in Pittsburgh, organizers should describe how they would convert their workshop to a hybrid or a virtual conference format if that becomes needed. I would also like to encourage you all to apply for a travel award to attend the Plant Biology 2021 meeting. More information about funding opportunities can be found on our website. Notice that the first two deadlines are in early December. We have an upcoming panel all about specialized metabolites coming up soon. This is a great chance for you to present your work in this series. All you have to do is submit a 7-minute video about your research. We will choose the top 4 videos to premiere live and then have all the speakers join in a panel discussion afterwards. These have been a really fun way to include more people and have more interactive webinars. If you work in this area, please send us a video. If you have a suggestion for a new panel topic, let us know in the chat box. Today's talk is the 22nd talk in our Plantae Presents webinar series. We've had so many amazing speakers this year. If you haven't already done so, please check out our archive to watch all the previous talks in this series. You can find information about each event on our plantae.org blog, and you can subscribe to our Plantae YouTube channel to get notifications when new recordings are posted. All right, those are just a few of the opportunities that are available from ASPB. I could probably go on for quite a while, but I know that you're here to hear our speakers. Be sure to follow us on social media and make an account on Plantae. You can also subscribe to our weekly newsletter, the ASPB Signal, to get more updates and opportunities directly in your inbox. If you need to leave today early, know that a recording of this webinar will be available within a few days. Each of today's talks will be 20 minutes. As you listen to each talk, you can add questions for our panelists in the Zoom question and answer Q&A section. Questions that are specifically for our panelists should be shared in the Q&A section to make it easier for our moderator to navigate. You'll notice that there's also a chat area open. This section is for you to introduce yourselves, say where you're from, and connect with your colleagues. Scott Hayes is with us today to moderate your questions. He currently works as a postdoctoral fellow at Wageningen University in the Netherlands. His research mainly sits at the interface between plant signaling networks. Throughout his career, he has researched light signals, temperature, salinity, drought, and how the clock all interact to shape plant development. He has been awarded several fellowships, including those from IMBO and Marie Curie, and he served as an assistant features editor for plant physiology. From next year onwards, he is looking for tenure track positions. So he would be happy to hear from you if you think he would make a good fit. We'd also give him a good recommendation here at ASPB. All right, with that, I'm going to hand it over to Scott to introduce our speakers and start the session. Thanks, Scott.
Thank you very much. I'm going to start off by introducing our first speaker, who is Carlos Ballaré. Carlos began his career in Buenos Aires, so during his master's, he actually performed a lot of research on shade avoidance and has really kind of laid the groundwork for that field. Following his master's, he moved to Oregon, where he started taking on a more agricultural, learn more about agricultural practices. But following this, he then moved back to Buenos Aires, where he really started heavily studying a lot of photoreceptors. So Carlos's work has really touched on UVB signaling, phytochromes, and cryptochromes. And more recently, also looking into how these photoreceptors affect plant defense. And I believe this is what Carlos is going to be talking about today. So yeah, maybe if Carlos then wants to start your talk.
Okay, thank you, Scott. Can you hear me okay? So, good. I'm gonna start the presentation. All right, can you see my screen now?
Yep, that looks great.
Okay, I'll still see some of your names on the screen too. That overlaps with some of the graphics. But anyway, so I would like to start by thanking the Plantae steering committee for the invitation to present our work here, and also to ASPB for sponsoring this series of webinars, which I think has been a fantastic idea and has been useful to keep our community more connected and more ensure more international representation. So my plan for today was to share with you some of the work that we've been doing to address a fundamental question in plant biology, which is how do plants optimize their growth and defense phenotypes under natural conditions when they are exposed to multiple sources of stress from the abiotic and the biotic environment. And in order to provide some ecological context, I'm going to use this image here. I used that before, so I apologize for those that have already seen this painting. This is certainly not a natural ecosystem, but it's an ecosystem that is full of interesting characters, which allows me to set the stage for the rest of the talk. So this is a painting by William Blake, and it's about Dante. Dante is the central character in the famous poem by Dante Alighieri, The Divine Comedy, or La Divina Commedia. And here's Dante, and he's trying to reach to the top of a sunlit hill, trying to get some illumination. But he's lost in a dark forest, and there he has to he has to deal with the monsters, with these three beasts. And of course, there is a lot of symbolism in the poem about the beasts, which I'm not going to cover today. But I think that you can imagine a seedling of your favorite shade avoiding species kind of trapped in the same situation. In the understory of a dense canopy, trying to reach for sunlight in a canopy gap. And at the same time, the seedling needs to be concerned about the threats posed by all sorts of consumer organisms, including herbivores, pathogens, and all kinds of consumer organisms. And the question is, how the seedling does that? How the seedling deals with all these compromises? And in fact, competitors and consumers are two of the greatest determinants of plant fitness under natural conditions. And the thing is that the growth responses that are needed to deal with competitors, that are useful to avoid being overtopped by competitors, call them shade avoidance, they tend to trade off with the defense responses that are required to fend off herbivores and pathogens. And what is the basis of this trade-off? Well, it could be just a simple allocation trade-off. So you have a limited pool of resources they can grow either to defense or to growth, but not to both. And that's it's also called the seesaw model, which is pretty popular in the literature these days, not in the form of the kitty cats, but in the form of this beam, in which growth and defense are represented in opposite sides of the beam as alternative sources for limiting resources. All right, we think that this is probably too simplistic. And last year we published a paper with Amy Austin, which we entertained this idea of a configurational conflict between shade avoidance and defense. And the point is that the phenotype that is optimal for shade avoidance can be maladaptive for defense and vice versa. So if you think of these elongated phenotypes with strong apical dominance and thin leaves, they're pretty good for shade avoidance, but they're not very good for defense, in which you probably would do better with a more branch phenotype, thicker leaves, and shorter stems. So in the presence of attackers, actually curbing growth is not a negative consequence of investing resources in defense, but it's actually a part of the defense. But on the other hand, curbing growth is not good for shade avoidance, right? And then you have the trade-off. Now, independently of the origin of the trade-off, the question is, how do the plants decide on resource allocation? How do the plants decide on what is the optimal configuration? And we're going to focus on that today. So in order to make decisions, plants need information. And as you know, light is a critical source of information for plants. So using specific photoreceptors, plant can obtain from the light environment information about a number of things. But in the context of this talk, the important thing is that you can, the plants can use light signals to detect the proximity of other plants. And that is because green leaves reflect a lot of far-red radiation. And this reflected far-red light provides a signal that can be detected by neighboring plants. So the way in which proximity perception works is pretty much like this. So if you're a plant and you're going without competitors, then you're going to receive a red to far-red ratio of about 1. And that's pretty constant. It's the same here in Buenos Aires, in Glasgow, or in Sydney. But if you have neighbors around, those neighbors reflect far-red radiation, and that reduces the red to far-red ratio. And when that happens, far-red inactivates phytochrome B by shifting the active form of the photoreceptor, which is PFR, into the inactive form. And because what this thing does is to inhibit elongation by inhibiting the PIFs. When phytochrome is inactivated, then the PIFs are able to promote elongation and trigger the shade avoidance syndrome, which is characterized by all these elongated petioles, stems, and things that allow the plant to be more competitive for light in dense canopies. Now, the molecular connections between the photoreceptors and the growth responses are becoming relatively well established. This is what I would call here in red the canonical PIF-auxin elongation pathway. But we are interested in understanding the connections between photoreceptors and some of these transcription factors with the morphological and physiological responses, which could be important for defense. So we try to understand how plants use information about neighbor proximity to make intelligent decisions about growth and defense. And the pattern that we and others have demonstrated is that plants grow under high density or in shade. They tend to have, this is associated with reduced defenses against herbivores and pathogens, and we try to understand the mechanisms. Some of the initial work was done by Miriam Izaguirre and Javier Moreno when they were students in the lab several years ago. But then we and others have demonstrated that when plants are grown under low red far-red ratios, they display a weak defense phenotype in bioassays. And also they express reduced levels of plant defenses. So this is a typical setup that we use to measure the effects of far-red on defense responses. So half of the plants are what we call the ambient light treatment, which are plants exposed to a high red far-red ratio. And then these other plants, the other group, is exposed to natural far-red to mimic the effect of neighbor proximity of reflecting far-red light. And then you can measure, do bioassays and measure your defense phenotype. So if you do the bioassays, you can see that in general, the treatment with far-red makes the plants more susceptible to herbivores and pathogens, and the attackers do better on the far-red plants compared to the ambient plants. So this is Manduca sexta, this is Podoptera, and here is botrytis, which is a fungus. And here what you get is that the plants with far-red are more susceptible to the fungus, and if you use a phyB mutant, in which you get this constitutive expression of the shade avoidance phenotype, what you get is constitutive susceptibility. Okay. Also, if you look at accumulation of plant defenses, defense metabolites, in general, when the plants are exposed to far red, the accumulation of these defenses is reduced, and there are many examples in the literature. So we want to understand the mechanisms. In order to get to the mechanisms, I need to introduce you to the Jasmonates, which is this family of lipid regulators which actually play a key role regulating growth and defense. So just for introduction, this is jasmonic acid. This is jasmonate disoleucine, which is the most bioactive conjugate, the more bioactive member of the family. And then there are a number of other compounds with different biological activity. And here is methyl jasmonate, which is the precursor that we normally use to induce defense responses or JA responses in plants. Jasmonate accumulation is induced in response to attack, is induced in response in herbivory, pathogen attack, mechanical damage, and then the jasmonates orchestrate the biosynthesis of defense metabolites. In addition, jasmonates are important growth inhibitors. So if you treat a wild-type plant with methyl jasmonate, you get reduced biomass. And if you do the same experiment in this mutant, which is a COE1 mutant that lacks a key component of the JA core receptor, then the effect on biomass accumulation is not present. Okay. So what is the effect of far red or phytochrome on jasmonate responses? And basically, we can look at the effects of far red on defense responses and growth responses. And there are many lines of evidence that low red far red ratios and in general the inactivation of phyB, for example, in the phyB mutant, tend to repress JA responses. And this has been shown at the level of the metabolome and at the level of the transcriptome. I'm going to show you a couple of examples. These are indolic glucosinolates, which are important defenses in plants of the mustard family. These are induced by methyl jasmonate in plants of the ambient treatment. But if the plants are exposed to far red, then the accumulation response is strongly attenuated. Also, if you look at gene expression, this is a heat map of a microarray in which we compare the response to methyl jasmonate between Columbia plants and phyB plants. And what you can see is that there are hundreds of genes that are induced in response to methyl jasmonate. And in 3/4 of this population of genes, the response is attenuated in phyB compared to Columbia. So this is what we call the cluster 2, which is the most representative, more abundant cluster in which the response is attenuated by the phyB mutation. There's another cluster here, cluster 1, in which actually you see a response to methyl jasmonate, but the response is actually stronger in phyB compared to Columbia, and I'm going to come back to that in a minute. Also, not just the response to methyl jasmonate, but also the accumulation of jasmonates is reduced in plants grown under shade conditions. These are experiments done by Anurag Agarwal with milkweeds and monarch caterpillars. And what you see here is that if you put the caterpillars on the plants, within an hour or so, you can get a very strong increase in the very strong JA burst, a strong accumulation of jasmonic acid in response to herbivory. But if you do the same experiment in the shade, the accumulation of jasmonate is reduced. So the general pattern is that phyB inactivation and shade result in reduced both JA accumulation and reduced response to the jasmonate treatments. We want to understand the mechanisms, and I'm going to spend the rest of the talk just discussing some recent progress in the regulation of JA metabolism by phytochromes. So I mentioned this cluster 1 before, in which the genes showed a stronger response in the phyB mutant compared to Columbia. And so we started to focus on this cluster because we thought that in that population of genes, there could be suppressors of defense responses under shade conditions, under conditions of low red far red ratio. So we searched multiple gene expression microarray databases and looking for genes that were are regulated by JA or related to JA, but were upregulated by far red treatments. So we explored a number of microarray experiments published by different people using different light treatments and different developmental stages, looking for genes that were JA related, at least by GO terms, which were consistently upregulated by far red. And we found like 30 or 40 genes in which there was a significant upregulation, consistent significant upregulation by far red. These are confidence intervals in the meta-analysis. This is work done mostly by my colleague, Carlos Mazza. And then this one here, this guy was our top hit, was consistently upregulated in all the microarrays that we were able to explore at that time. So it's the name of the gene. And this gene encodes sulfotransferase as SOT15 or ST2A. And as you may know, sulfation is an important mechanism by which the biological activity of many small molecules, including hormones, is regulated in mammalian systems, but their role in plants is not well characterized. So ST2A in this evolving map of Jasmonate, and we call this an evolving map because different compounds are being discovered and different reactions have been discovered in recent years. And but ST2A in vitro can use hydroxy-JA and then form this JA sulfate, which is an inactive compound. So we thought that perhaps what far-red is doing is to promote the inactivation of JA through this metabolic pathway. So first we confirmed that far-red or that this gene was strongly upregulated by far-red because most of the data that we had were from microarrays done under completely strange conditions, or I mean different conditions that would not necessarily were designed to mimic the effects of neighbor proximity. So we tested for the expression of this gene in our far-red conditions, and as you can see, this gene is upregulated by methyljasmonate, but the upregulation is much stronger when the plants are exposed to far-red, and this is work done mostly by Guadalupe as part of her dissertation. So we then determined that far-red upregulates ST2A through the PIFs, ST2A transcription. The PIFs directly bind to the ST2A promoter and control its activity. And so when far-red inactivates phyB, the PIFs promote the transcription of ST2A. This is collaboration with Christian van Hauser and Anne-Sophie Fiorucci from Lausanne. Then if you do the genetics, you can see that P4 plays a role in the upregulation of ST2A by far-red. But actually the central player is PIF7. So if you lose PIF7, then the expression of ST2A by far-red is greatly attenuated. In this triple 457 mutant, you basically don't get any induction of ST2A expression by far-red. So then we wanted to do a complete characterization of the effects of far-red on the JA metabolism. So we treated our plants with methyljasmonate, then we harvested at different times and measured the abundance of all these compounds. And this is mostly done in collaboration with Michael Reichelt in Jena. So essentially what we found is that far-red downregulates all the JA pools. So this is, so the red bars in general are shorter than the white bars, and that involves jasmonic acid and JA-ILA and all the conjugates. But there was a clear exception to the pattern, which is this JA sulfate, which was actually strongly upregulated by far-red, and that coincided with a strong upregulation of ST2A transcription. So if we go back to this slide that I mentioned before in the work with milkweeds, it's well documented that plants in the shade induce reduced levels of jasmonate, and then we wanted to know if this effect of shade could be related to phytochrom and to the effect of far-red on jasmonate inactivation via ST2A. And to do that, we used the tools available, genetic tools available in Arabidopsis, and we replaced the caterpillars by mechanical wounding. And then we did this experiment in which we wounded the plants and we harvested for different JA pools to measure different JA pools. And essentially, we found three important things. And the first one is that far-red actually reduces the accumulation of jasmonic acid, and that is pretty similar to the results that these other guys have obtained under natural field conditions. And then this reduction in JA accumulation coincides with a strong accumulation of JA sulfate. And third, that both things, the effect of far-red reducing the accumulation of JA and the accumulation of JA sulfate were completely dependent on ST2A. So in an ST2A mutant, you don't see this effect of far red, and then the accumulation of JA sulfate is completely missing. So that tells you that although there are like 21 genes encoding for putative sulfotransferases in Arabidopsis, ST2A seems to be the only one that is responsible for generating this JA sulfate metabolite. Okay, and then final question is this effect of far red promoting inactivation of JA functionally important? And then what you can do is to, again, use genetic tools that measure different JA outputs or responses in the area of plant growth and plant defense. So if you look at JA markers, these are all typical markers of JA responses like LOX2, MIG2, BSP. So in wounded plants, these markers are strongly induced. And, but as you can see in Columbia plants, the effect of far red downregulates the expression of these genes, which is the typical pattern that you would expect. However, in the ST2A knockout, that effect of far red is completely gone. As you can see, there is a strong genotype by light interaction for all these markers. And actually, in wounded plants, there is a clear, in the RNA-seq, there is a clear overlap between the defense-related genes that are downregulated by far red and those that are upregulated by the ST2A mutation. So these are the Go descriptions for these genes, and they are all related to defense and pathogen defense and responses to JA. And that clearly suggests that the effect of far red reducing the expression of plant defenses is mediated by ST2A. So if you do the bioassays, I mentioned before that the plants that are exposed to far red tend to be more susceptible to herbivores, and this is certainly the case here with Spodoptera littoralis. As you can see, the far red plants in the Columbia genotype are much more severely damaged than the control plants. However, in the ST2A knockout, you don't see that effect of far red at all. And also, same thing with botrytis. I mentioned that far red makes the plants more susceptible to botrytis, and this is certainly the case here too. When the ST2A knockout or a triple PIF knockout in which there is no induction of ST2A transcription, then this effect of far red is missing. And interestingly, in overexpressing ST2A lines, which were developed in collaboration with A.B. Kuhl, then what you get is a constitutive susceptibility. So what about plant growth? And this is the last piece of data I'm going to show you today. So these plants, the ST2A knockouts, they look pretty normal under ideal growth chamber, non-stressful conditions. As you can see, they have a pretty normal rosette, and they show a decent or a pretty normal shade avoidance response, which is characterized by elongated petioles and also more hyponastic leaves, which are very, very similar between Columbia and the ST2A mutant. However, if you stress the plant or if you put the plant a little bit of methyl jasmonate, then the shade avoidance response is clearly compromised. You get a reduced petiole elongation, and the hyponastic responses is also attenuated. So our conclusion is that a key function of ST2A is that when the priority is to avoid shade, is to release the growth brake that is created by the jasmonates, and thereby allow the plant to fully express its shade avoidance phenotype. So going back to our original question, how do the plants choose the right configuration or the right resource allocation pattern? This is our model in which far red attenuates defenses and facilitates shade avoidance by increasing ST2A transcription and the sulfation of jasmonate. So that diverts JA precursors away from the formation of the bioactive conjugates and into this metabolic sink that is JA sulfate. And that this work suggests that the PIFs provide a direct link between phyB and jasmonate signaling. So if we go back to our painting, here Virgilio is telling Dante about your fears, your path to heaven, to illuminated heaven. But if you replace Virgilio by evolution, probably what natural selection has told the Arabidopsis is that, hey, when far rate is high, that means that your neighbors are getting pretty close. So you better sulfate your Jasmonates and try to grow to the light as soon as you can. And a couple of additional take-home messages. One is that obviously there is a lot more to shade avoidance than just the elongation. The PIFs are key regulators of Jasmonate and defense responses, and I don't have time to cover that today, but it looks like there are lots of feedbacks and interactions between pathways that allow the plants to actually optimize their configuration in complex environments. And finally, like plants like mammals, and they seem to use sulfotransferases to regulate the activity of hormones and therefore regulate phenotypic plasticity. And with that, I'm going to close. I guess I mentioned everybody in the talk that participated during this project. I need to also acknowledge some other people that was not involved in particular experiments, but was important during our discussion over the years, like Rona Pierik, for instance, and then a couple of new students that have joined the lab, Michaela Lici and Anna Medina. As I mentioned, most of the work on ST2A was done by Guadalupe Fernandez-Miranda. I need to mention my funding too, particularly CONICET and FONCIT, and in this particular case, the von Humboldt Foundation was very instrumental in facilitating the collaboration between our group and Jonathan Gersherson's department in Vienna. And thank you for your attention. I'd be happy to take questions if we have time.
Okay, great. Thank you very much, Carlos. It was a very interesting talk and a very important reminder that we need to kind of think about what plant, like from the plant's point of view, what's going on is, they've got to worry about these other things as well. So I'll just have a look if we've got any questions coming through. There's something that I would actually like to ask you. So my main question is, why do plants so heavily downregulate their defenses in response to shade? I mean, you seem to, so you show that they can only mount a reduced shade avoidance response if they don't sulfate their Jasmonates. But this is mainly manifested in reduced hypernasty, which to me seems a very low cost shade avoidance response, just moving your leaves upwards. So what is the question more like, what is the real, what is this energy trade-off? Like, why did they do this?
Well, it is two things. I mean, it's low cost in terms of probably the carbon or the nutrients that you need to make this hyponastic response or petiole elongation response. But it's costly in terms of the risk that if you know under pressure from a grazer, you probably are better off by keeping your leaves down, and then you're not getting yourself exposed to the attack of herbivores. So I think that part of the cost has to do with the fact that growth reduction is actually a part of the defense, but it's not very good for shade avoidance.
Okay. So we've got a question from Nathan Havko. So do you expect a significant overlap in the effects of shade avoidance and heat-induced hypernasty through phyB?
Yeah, this was, I guess, if I understand the question correctly, is whether the same kind of interaction would be between heat-induced hypernasty and hyponasty induced by far red. I guess I would expect a pretty similar kind of interaction between, we are talking about the same players, which are phytochromes and the PIFs. And so we haven't done the experiment, but I would expect a similar kind of crosstalk there.
Okay. I think we have time for one more question. Oh, so this is so it's maybe also leading into the following talk as well. So we have a question from Dimitria Angelapoulou, sorry for the pronunciation. Dimitria asks, are the phytochromes linked with the photosynthetic mechanism? So how does this kind of, is there an overlap there?
Yeah, I think that we the people that work in photoreceptors, we try to forget all the stuff that has to do with chloroplast, but of course, chloroplasts are important regulators of plant defense as well. So we haven't got into that, but certainly there's a whole world to be discovered there, which I think we in the photomorphogenic field, we tend to not to think about photosynthesis too much. But there is evidence that the chloroplasts control defense responses, and also the chloroplasts are the site of biosynthesis of Jasmonate. So I would expect a lot of interaction there. We haven't got into that.
Okay, well, I think that maybe is a good point to leave it and then move on to our next talk, which is focused on photosynthesis.
Okay, right. Okay. Thank you.
So thank you very much, Carlos. So our next speaker is Haim Trebes. And Haim began his PhD in the Hebrew University in Jerusalem. And there he was instrumental in characterizing an extremophile alga known as Chlorella ohadii. I think I pronounced that correctly.
Mm-hmm.
I've been reading a little bit about this extremophile, and it's quite incredible. It seems to be able to survive in conditions from sub-zero to 60 degrees Celsius with extreme light and very little water. So Haim's research has now moved to Germany, where he's at the Max-Planck Institute in Gulm. And there he's studying just how it is that this alga can survive in these conditions. So with that, I'd like to turn you over to Haim, and so here's the talk.
Okay, can you hear me well? Okay, good. Okay, thank you, Scott. Okay, and thank you for this kind invitation and for organizing this wonderful series of talks. Today, I'm going to try to describe why and how can we harness the potential found in algal metabolic diversity to improve our understanding of plant biology and as a first step towards plant yield improvement. So I'm going to skip beyond a bit only Chlorella ohadii. I will start by describing how big is the problem or how large is the challenge, depending how you decide to look at it. Then I will provide a couple of case studies which will demonstrate how limited is our understanding of this potential as a resource. And then I'm going to get to what I'm doing today and what I hope to do once I lead a group of my own to fulfill this potential. And I'd like to start by the motivation. And here I think that not enough people are aware of how close we may get to a food crisis even during this century. The predicted increasing global population, together with the increase in our living standards, may require agriculture to more or less double food supply by the middle of this century. And as extreme as this may sound, this is not the first time that agriculture has been requested to do that. In the second half of the previous century, in the so-called Green Revolution, agriculture was actually already successful in doubling food supply, relying about 50-50 on improvement of growth through breeding and improvement of interface, namely irrigation and fertilization techniques, soil density optimizations, etc. But can everyone see this small plateau here in food supply? Good, because it's going to be important in a couple of slides. So like I said, agriculture was already successful in doubling food supply, only that this time it looks like it's going to be even much harder. Since we already allocate today much less land per capita for agriculture, a trend which is even expected to become worse in the next few decades. In addition, the predicted increase in global population may pose even more severe limitations on the availability of fresh water for agriculture. And that's even before saying one word about climate change, which has very direct effects on the productivity of the agricultural sector. So whatever progress has been made in the last 2 decades has been more or less abrogated by this trend, keeping us around the zero balance, which is better than a negative balance, but still. And now I'd like to even go back to that plateau I mentioned earlier. Well, apparently and unfortunately, it seems to be quite a stable trend in most major crops worldwide. And more researchers and agronomists are trying to find some explanations for that. And they frequently come back with the answer that many of our current approaches are starting to become saturated. So in order to better explain what they're talking about, let's spend 20 seconds on what is plant yield potential, which is a basic multiplication of the following three elements. First, the amount of light that can be intercepted by a given field, for example, throughout the growing season, times the photosynthetic efficiency, which is quite straightforward, and times the harvest index, which is the proportion of biomass invested in the crop into whatever it is we'd like to harvest, whether it's fruit, seeds, or tubers. Now, the first and the last one definitely improved during the Green Revolution. The last one through breeding for optimized harvest index, and the first one through denser canopies, or what is termed early canopies, with fully expanded leaves early during the growing season, but they both have been recently estimated to reach very close to their theoretical maxima. Photosynthetic efficiency, however, has been much less exploited, and it is also considered to be highly attractive because first, it is expected to be conserved among most major crops, and also it can rely on a vast knowledge database of a century-old photosynthetic research. And indeed, in the last 2 decades, publications related to photosynthetic improvement of crops has experienced quite a golden era, as you can see in here. And one of the leading groups in the world in this field took advantage of the progress made in modeling tools and computational power to try to predict where are the major losses of photosynthetic efficiency and where should we focus our future efforts. And this is how typical biomass yield prediction looks like for a C3 or a C4 crop, and these are the different losses. And most of the work in recent years focused either on improving the light harvesting of these plants or increasing the CO2 concentration around rubisco, and by that minimizing the need for photorespiration, or generating alternative routes for photorespiration, which are less wasteful in terms of energy. With the exception of very few single Kelvin Benson cycle enzymes, much less work has addressed what I call photosynthetic metabolism or photosynthetic investment, despite its high predicted value. In addition, in any of these models, regardless of the final results that can be largely dependent on how you define your inputs or outputs for these models, algae always scored higher compared to both C3 and C4 plants, which at least to my opinion, puts a very clear spotlight on their immense potential to improve plants in the future. And I would like to target this potential focusing on the photosynthetic metabolism. And algae are quite a diverse organisms that can be found in various niches, from freshwater to wastewater, from marine environments to soil crust, even desert crust. Much of the work I'll present here today was inspired by one highly capable desert alga, which has been recently isolated. And like you heard, this alga is called Chlorella ohadii. It's one of the late Professor Itzhak Ohad, one of the godfathers of photoinhibition and photosynthesis research, which I was privileged to work with in my first year of PhD. And I think that the most prominent trait of this alga is its remarkable growth rate. Generation times as short as 2 hours under optimal conditions comparable to yeast, which to the best of our knowledge is the fastest growing ever reported photosynthetic cell, at least eukaryotic. In addition for being such a fast grower, it demonstrates what seems to be quite a unique growth pattern if you compare it to classical microbial growth curves, as you can see here with these green curves and this multiphasic behavior, both in low light and also in very high light. And trying to integrate the physiological data we collected during this experiment, it looks like the cells are shifting between very high photosynthetic rates to very high respiration rates, as if they are shifting between two metabolic states of accumulating photosynthate and then utilizing it, and then accumulating again and so on. One more thing I'd like you to note here is these very sharp stops in their growth, almost not biological in their nature, indicating that the cultures are highly coordinated around this inflection point. So in order to establish that these are indeed metabolic shifts, and also to try to point out some metabolites or molecules that can be involved in the signaling of this growth rhythm, we perform metabolic profiling of several time points along this growth curve. And the first thing that became clear to us was that this metabolic profile clustered mainly according to the growth phase and only later according to the light level, even though it was completely different. Further supporting the idea that these are indeed metabolic shifts, that's what distinguishes the different phases, is their metabolic signature. In addition, this analysis also allowed us to identify some possible molecules or metabolites that can be more predictive for the response of the entire metabolome, and these ones got the highest scores and became a very clear candidate for more signaling molecules. And indeed, external provision of some of them to the running cultures of Chlorella ohadii had very clear positive or negative effects on this metabolic flexibility in the growth of these cultures. But I still didn't answer the question of why is this growth pattern favorable for this Chlorella? What do they have to benefit from performing this growth arrest in the middle of the growth curve? I'm going to ruin the surprise. We still don't have a good answer for that, but we do know that if we manipulate the conditions in one of several ways to eliminate this growth arrest, the cells reach lower final cell densities, even under higher energy or carbon input, like in this specific example, meaning that something happens within this growth arrest that allows the cells to reach higher final cell densities and eventually grow better. And trying to project this to the desert crust from where they were isolated, we should bear in mind that water are only available in the crust for about 290 years thanks to dew, coinciding with light for about 1 hour or less each morning, and leaving a very short and limited window of opportunities for positive photoautotrophic growth. So we think that what we see in the bioreactors is actually a reflection of this unusual diurnal cycle, with early morning dedicated for photosynthetic accumulation, followed by a long, dry, and inactive day, which we cannot simulate in our bioreactors, and then upon rewaking in the following night, the cells can utilize the photosynthesis for growth and respiration. Now, as students in microbial ecology, we are usually taught that you can be either an excellent extremophile or a very fast goer under optimal conditions, but not both. So what's even more intriguing in the case of this green alga is its remarkable resistance properties on top of its fantastic growth. Now, if you take, for example, its closest relative from freshwater, which is called Chlorella sorokiniana, and you expose it to twice full sunlight levels for 24 hours, you get this bleach morphology, even though it is considered to be quite a high light tolerance strain. Now, if you perform the same experiment with Chlorella ohadii, the difference is striking. Not only is not harmed by the excess light, it even grows better compared to low light, performs higher photosynthetic rates, and also demonstrates higher affinity towards inorganic carbon. And in a paper we previously published, we associated this extreme resistance to an altered function of these alga photosystems, and specifically photosystem 2, protecting it from damage, but I'm not going to get into the details due to the time constraints. But anyway, we had in hand, for the first time, a photosynthetic metabolically active cell that does not literally burn at 3000 micro-Einstein. And for the first time, we could characterize downstream cellular responses under these extreme conditions. So we decided to perform quite a wide systems analysis of this alga response to extreme illumination. And one of the things we had to take into account, at least in this organism, was that long before processes like changes in gene expression or de novo protein synthesis could take place, the responses at the level of growth and photosynthesis were already quite dramatic, meaning that at any given moment in low light, Chlorella ohadii cells possess the enzymatic and metabolic capacity to utilize an abrupt 30-fold increase in their energy input, which is first quite remarkable, and second had very clear consequences on our experimental design. So we came up with the following sampling plan, trying also to capture the very early component of the cell response to extreme illumination by following their gene expression before, 15 minutes and 2 hours after the shift from low light to high light, and also in continuous highlight as a stable point for some of these changes. In addition, we followed the response of the redox response of their proteome before, 3 minutes after the shift to highlight, trying to capture also the very early post-translational modifications under these highly reducing conditions, and also in continuous highlight as a saturation point for some of these changes. In addition, we also followed the levels of primary metabolites and lipids in similar time points, and we took several indices related to their physiological responses, growth, photosynthesis, respiration, and morphology. Naturally, I will not be able to cover most of this work in this very short presentation. Instead, I chose to focus on one biological story which will demonstrate some of the messages I'm trying to convey here. And this is related to Chlorella ohadii cells' metabolic capacity to oxidize the excess redox, which is usually accompanied to a high light treatment in a photosynthetic cell. And this actually started by quite a surprising observation we had when we followed the levels of NADPH in this algae using a dual pump. And we found out that contrary to what we know from model organisms, and very shortly after a shift from low light to high light, within 15 minutes, Chlorella ohadii cells demonstrated actually lower levels of NADPH. And this meant that they had to activate quite fast several processes related to NADPH shuttling from the chloroplast and its oxidation in the cells. And given the very early timescale of this response, we focused on the redox proteome to find some answers for that. And there we found several cases of unique redox-dependent behaviors in key enzymes related to NADPH metabolism in this alga. And the first one was the plasmid malic dehydrogenase, maybe the best characterized protein in redox biology, which in this case showed no redox response. Now, importantly, in vitro studies of these enzyme variants lacking the redox sensing cysteines demonstrated higher maximal activity. In addition, we did not find any redox response in the previously characterized to be redox responsive cytosolic aldolase, perhaps representing a similar example in terms of activity. And on the other side of the story, we found the previously uncharacterized redox response in a cytosolic non-phosphorylating NADP-dependent GAPDH, which has been implicated in the past to play a role in arabidosis in shuttling of redox from the chloroplast under conditions of very high photosynthetic rates. And we also found that a redox response in a previously uncharacterized variant of an NADP-dependent malic enzyme. And so far, only 4 variants of this enzyme have been found to be redox responsive. So all of this pointed to quite an efficient cellular machine for NADPH oxidation, which should have resulted in increased respiration rates. And indeed, shortly after the shift to highlight, we found a significant increase in the respiration rates of the Chlorella ohadii cells. Now, we also wanted to try to explain this unique redox-dependent behavior, so we decided to examine the sequences of these enzymes in Chlorella ohadii and compare them to the databases. And there we found several cases of unusual cysteine patterns, either by the absence of very highly conserved cysteines or the appearance of new cysteines in very unusual positions, which were otherwise highly conserved. Now, interestingly, this feature also was shared between Chlorella ohadii and other Chlorella species, including those that are far less resistant than Chlorella ohadii to extreme illumination. And in addition, this also happened as a general background of a higher cysteine count in Chlorella ohadii genome, including in functional aspects that can be highly related to the response to extreme illumination. So to conclude this part, I would say that as a fact, Chlorella ohadii cells demonstrate very efficient metabolic capacity to oxidize this excess redox, this NADPH, and we associate that with unique cysteine patterns, with a unique redox-dependent behavior in key enzymes related to NADPH shuttling and oxidation in these enzymes or in these cells, and we associate that with unique cysteine patterns in these enzymes, a feature which, like I said, is also shared by other Chlorella species. So our understanding of this story is that on the way from being a fresh or a wastewater alga like the rest of the Chlorella relatives to become established as a desert organism, the stepping stones included first the development of such an efficient metabolic capacity to oxidize the NADPH as a stable ground for the development of such a robust photosystem to rendering them more resistant than other Chlorella species and allowing them to be established in the desert. And think about it, just like no reasonable engineer would install a V8 engine on a Dacia body, nature, evolution, if you will, would not allow the development of such a superb photosystem too on a metabolically incapable or less capable cell. And this brings me to the last part of my talk, in which I'll try to discuss what I'm doing today and what are my future plans for this project. And clearly, this alga provided us with inspiration to raise questions like, what makes at least a photosynthetic resource fast? What sets the upper limits for growth for photosynthetic cells? And what makes some of them grow faster than others? Now, trying to answer these kind of questions using comparative approaches like comparative genomics, transcriptomics, and to some extent even proteomics typically failed because we usually ended up with too long lists of candidate genes or candidate proteins that may be involved or may play a role, and we eventually ended up with potential. And I wanted to tackle these questions by directly approaching the metabolic phenotype, namely the kinetics of all of the possible metabolic reactions, starting from the first one in photosynthetic cells, carbon fixation, and downstream. And then once we find an interesting difference, to approach that mechanistically. In order to do that, we designed a labeling flux trace in which we took several algae with different growth rates, fed them with certain CO2 using a tailor-made microfluidic mixture, which we designed in order to be able to resolve the kinetics of the very fast reactions of photosynthetic metabolism. We then follow the labeling to different carbon cellular pools and to different metabolites. And this allowed us to successfully identify metabolic bottlenecks in the slower growing algae, which are overcome by the faster growing ones, and also specifically Chlorella ohadii. Just to provide a few brief examples, we found very interesting differences in the RUBP regeneration part of the Kelvin-Benson cycle, in the flow of carbon from the Kelvin-Benson cycle to lower glycolysis, and also in an aplerotic carbon flow into the TCA cycle and protein synthesis. And I think that for these two of these differences, we have something promising in hand because they are found in very conserved part of photosynthetic metabolism, and they are also considered by many to be classical metabolic bottlenecks in plant metabolism. And I think that by now you should not be surprised if I told you that in enzymes found in key positions in these pathways, we found unique substitutions in very key residues, both in regulatory domains or in active site domains, when we compared the algal variant of these enzymes to their plant homologue. Only that this time, instead of working on a very long list of candidate genes or proteins, we can work now on a very focused list of cases where this probably made a difference at the level of the metabolic phenotype, and now we can target them mechanistically or genetically. And this takes me to the last slide of my talk, in which I try to discuss what are my future plans for this project. And there are three main aspects for my future plans. The first part is the part where we cache out all of the insights from this systems analysis and target the specific mechanisms involved. Here I'd like to take advantage or use existing tools developed for Chlamydomonas, and already with some preliminary success for transformation system we have for Chlorella ohadii in collaboration with a group of Professor Peter Edelman in Berlin University. In addition, we'd like to perform quite a wide enzyme activity assay of over 40 enzymes in central metabolism, comparing the different algae, and by that providing some kind of a validation for our flux estimate, and also the ability to raise new targets for analysis. In addition, we'd like to more deeply characterize the biochemistry of these enzymes, at least the algal variant of this enzyme, focusing on the catalytic rates and also on their regulatory properties, and by that try to tailor a metabolic phenotype to the biochemical mechanism. In addition, we'd like to upgrade our experimental setup, focusing on its automation and sensitivity in order to be able to accommodate a wider analysis of algal metabolic diversity. And this takes me to the last point, which I probably mentioned already more than once, that we'd really like to expand the pool of algae we explore and to really explore algal metabolic diversity. I personally have very high expectations for algae from intertidal zones and from polar zones for various reasons. I'll be happy to discuss later. And also, most of the data I presented here today was obtained using what we call standard conditions, at least in terms of illumination and temperature. And I think that this is potentially a very powerful tool to predict how would algal metabolism, including key ecological species like diatoms or coccolithophores, will respond to change in environment. By manipulating the growth or labeling conditions, we can try to predict how would these key ecological species respond, for example, to climate change. At this point, I'd like to thank all of the people involved in this project, both in the MPI here in Bonn and the Hebrew University, where I did my PhD, our collaborators here in Germany, in the IPK in Gazaslieben, Marburg University and Berlin Humboldt University, in the Weizmann Institute, our funding, and you for listening. Thank you.
Okay, thank you very much. So yeah, it's clearly a well, it's an incredible organism. Yeah, kind of the first question which really springs to my mind. This organism is so efficient at photosynthesis, and it can survive in extremely high light levels. Why is it that not all plants are always prepared for this? Like, what is the cost, or similar to as Carlos was explaining, like what is the cost for having this protection? They can't all be benefits.
Okay, so there are two ways to address your questions. The first part is, if you are susceptible, the cost to be able to withstand this kind of levels of illumination would be the reproduction of all of the susceptible proteins or enzymes which are sensitive to light. One is more known than the rest, and it's called the D1, the major protein in the structural protein in photosystem 2, which in the case of Chlorella ohadii is just less sensitive. It's not that they are producing it faster. We showed that even when we block the novel protein synthesis, it is not degrading as fast as it is in a normal plant, let's call it. Okay. In addition, I think that it's not the cost of having the entire metabolic machinery for utilizing 3000 micro Einstein is quite high. Okay, to be able to reach lower levels of redox in the cell in response to very high light means that you have to hold quite serious levels or amounts of enzymes which are dealing with just oxidation of excess redox. Now, if you are living in a world where a highlight can just come every morning, then it makes sense to invest that, especially if you have 1 hour of activity each morning because you are dry throughout the rest of the day. For a normal plant, let's call it not in the agricultural field, but in nature, this is usually not the case. In agriculture, maybe the challenges, or in a field, the challenges can be even maybe more similar to what Chlorella ohadii has to experience compared to nature, but depending on on many things, the crop, the environment, etc.
Yeah, I mean, you showed that. Yeah, I was just very surprised that you showed that even under very mild conditions, it could still survive extremely well.
Yeah.
So yeah, it's interesting. I have a question from Mary Williams. So are there other isolates of this species from less intense environments? So what are the habitats of its closest relatives?
Yeah. So most of its closest relatives come from actually from let's call it habitats which are favoring high performance and fast growth. Okay, most of its relatives come from, for example, from wastewater treatment facilities where there is enough nutrients and the conditions are supporting fast growth. And actually, we think that Chlorella ohadii found its way into the desert crust through being re-inoculated several times from one of these wastewater treatment facilities to the point where one event or more than one event was resistant enough also to deal with desiccation and the other stressors in this habitat. So I would say that most of its relatives, I'm not talking about the entire genus of Chlorella, but Chlorella sorkiniana, Chlorella purinoidosa, Chlorella vulgaris, the ones which show a higher similarity to Chlorella ohadii are mostly found in these rich environments, let's call it.
Yeah, thank you. I think we might have to call it a day there, and we'll try and answer some of the questions that people have posed we didn't get to actually talk about in the talk. I don't know if Katie is wanting to share anything else.
Thank you all for joining, and we hope to see you at our next event. Bye, everyone.
Okay, bye. Thank you.